Okay, good morning. We will start timely. I think many people are still on their way because this room is very far away from the main building. However, we must keep the timing. I would like to say very short inflammatory remarks. First of all, thanks to my co-chair, Professor Pasquale Maffia. And I am Francesca Levi-Schaffer from the Hebrew University of Jerusalem, Israel. And we have been involved together in the creation of the IMUPHAR, that is the Immunopharmacology group at IUPHAR. And we are very active, and we hope you will also like to join us. The immunoinflammatory responses, as you know, key a play role in the main pathogens leading to morbidity, mortality, such as in autoimmunity, in cancer, in allergies, in autoimmune and metabolite disorders. Our research tries now more to understand what drives this response, but also hoping to be able to downregulate them, if not to stop them, to understand how they are regulated. Therefore, we decided to have this symposium in which we try to investigate different inflammatory diseases, in which basic scientists from immunology, pharmacology, biochemistry, and the clinicians should be involved. Therefore, I will already start to invite the first speaker. I mean, is my co-chair, Professor Maffia. We invite the first world-renowned speaker.
Okay, good morning, everyone. The first speaker, it's a pleasure for me to introduce to you Professor Alberto Mantovani. He's a professor between the Humanitas Institute in Milan and the Queen Mary University in London. Alberto is a world-class leader in the field of immune response in cancer, and we'll discuss today about where we are with targeting immune response in the pathology. Thank you, Alberto.
Thank you. Yes, I want to thank the organizers for the privilege to be here. I will focus on cancer, but before entering cancer, I will share with you a general view of some aspects of inflammation. And of course, inflammation is downstream of sensing infection. I don't know if yeah, here it is. Infection, tissue injury, homeostatic imbalance, and here are the markers that we use. C-reactive protein is a paradigm for them. And this is a molecule of ours that we cloned that is a distant relative of C-reactive protein. And these are generally represented as a collection of weird molecules. And Cecilia Garlanda and myself in this review in New England Journal try to make sense of that. So here are the sensors of innate immunity that trigger inflammation, which are strategically located on the cell surface, in the exosomal compartment, and in the cytoplasm. And here are the professionals, myeloid cells in particular, as well as non-professional cells. And this is where all the hype is, the cellular arm of innate immunity. But there is a humoral arm of innate immunity, which is generally forgotten, and it's made of a bunch of apparently weird molecules. Our connection here is with the cloning of the first long pentraxin, PDX3, a distant relative of C-reactive protein. And we think that there is a logic here, because these molecules, in essence, work like antibodies, because they bind pathogens, for instance, they bind or tissue damage, they activate complement, they have opsonic activity, and they regulate inflammation. And the production of these molecules is downstream of a cascade which involves with negative regulators, which involves primary inflammatory cytokines, downstream mediators, for instance, chemokines, and then the liver, which is a major source of some of these acute phase protein. Acute, by the way, acute is a misnomer because they are elevated in chronic conditions. And, but the liver is not the sole source of these molecules, because myeloid cells in particular contribute to local production of complement components and some of these molecules. And the logic of the acute, in quotes, phase response is on the one hand amplification of humoral innate immunity and defense, and on the other hand, provision of tools, of building blocks for tissue repair and for regulation of tissue repair. And my own connection, and of course myeloid cells are central players, and my own connection to the field stems from my interest in myeloid cells and macrophages in tumors in particular. I want to emphasize the key contribution of Bob Evans, because he's the one who originally showed that macrophages in tumors are bona fide normal cells. They are not tumor cells in these guys. And he and Bob Evans originally showed that these cells could kill tumor cells. I had the privilege of working with Bob Evans and Peter Alexander, but my views diverged because I found that in human metastatic tumors, in human real cancer, and in mouse metastatic tumors, actually macrophages served as helped promote the tumor growth, and that led to one of the very early descriptions of chemokines, because we discovered them CP1, CCL2, as a tumor-derived chemotactic factor. And the view that emerged at the time is that of a symbiotic relationship between myeloid cells and cancer cells, whereby cancer cells attract macrophages, and macrophages serve as tumor cell little helpers by providing growth factors, by promoting angiogenesis, and by taming of adaptive immune responses. And we will get back, and I will get back to that in a moment. At the time, there was just one macrophage. But of course, we are now doing single-cell analysis, and the next slide is single-cell analysis done by Vasily Kandinsky. And here is the classically activated macrophages, the one described by Bob Evans and Peter Alexander. An alternative form of macrophage activation was originally identified by Simon Gordon in Oxford, and we contributed to the description of this. When we did the first, and we is Shubhra Biswas in my lab at the time, when Shubhra did the first transcriptional profiling of tumor-associated macrophages, we found a distinct phenotype that did not really match the two extremes. And so it was an early indication of the diversity. And we'll get back to single-cell analysis later in the talk. We have made progress in defining the molecular basis of macrophage activation and deactivation of tolerance. And there are two messages here. One is that we have made progress in understanding molecular mechanisms. And the second message is that what we have long known as tolerance and responsiveness indeed is a distinct transcriptional program, and the general significance of this is protection of the host against excessive, uncontrolled inflammation. And then early on, many people did observations that hinted to some form of memory in innate immunity. And Mihaly is the one who worked out much of the mechanisms and implications and coined the term trained innate immunity. And training is centered on mature macrophages. So again, Simon and I had some early data on this. Macrophages, myeloid precursors, epigenetic reprogramming, and then enhanced microbial killing, production of cytokines, production of anti-antibodies, these fluid phase pattern recognition molecules, and activation of adaptive immune responses. Now we have a better sense of the interplay between myeloid cells, macrophages, and tumor cells, and the diversity, and this is our own drawing, and these are Kandinsky drawing. You make your choice, which you prefer. And of course, these cells contribute to genetic instability, to the buildup of a niche, a metastatic niche, remodeling of the tissue and angiogenesis, and suppression using a variety of tools, suppression or inappropriate skewing of adaptive immune responses. Macrophages have served as a paradigm for the connection between inflammation and cancer. And we like to think, and when I say we, this Fran Bolquill, at William Harvey, myself and others, and Antonia and Paola, we like to think of this connection as consisting of two pathways: an extrinsic pathway whereby inflammatory conditions increase the risk of developing cancer. For instance, colorectal cancer and inflammatory bowel disease is an example of that. And an intrinsic pathway whereby genetic events orchestrate the construction of an inflammatory microenvironment. And as I said, macrophages have served as a paradigm for that. At the time we had this review in Nature, many years ago, this part was not there. So humoral innate immunity was not there. And then in my lab, Eduardo Bonavita did the first time genetic studies on this molecule of ours and complement, and found that humoral innate immunity plays a role in the orchestration of the tumor microenvironment. So complement is a double-edged sword. Of course, you can kill target cells with complement, and on the other hand, can drive tumor-promoting inflammation. But we like genetic evidence in mice and humans, and we realized that there was no genetic studies on pathways of complement activation and actual effector mechanism using primary carcinogenesis. This is another aspect that we like, this primary carcinogenesis. So what we set out, and when I say it's Elena Magrini and again Cecilia Garlanda, we set out to do systematic analysis of the pathways of complement activation, the three pathways of complement activation, and effector mechanism using gene-targeted mice and primary carcinogenesis, sarcomagenesis, I should say better. And this is the key. There is more. We have more unpublished data. But in essence, we confirmed the early experiments done by Eduardo Bonavita in my lab. So the C3 mice are protected. Interestingly enough, MBL, so the lectin pathway here is a key driver of complement activation. And unexpectedly, it's not C5a, the effector pathway, but it's C3a. And these are two. This is a bona fide receptor, and this is a unconventional C5 receptor, no phenotype there. So then we did what, and I go quickly, this is the data and unpublished and more unpublished data, and C3 deficiency was associated with reprogramming of leukocytes, with increased antigen presentation. Then you come up with a signature, ask the question whether the signature is associated with prognosis. You go into TCGA, you find that this is the case, but then we like to do studies in our patients, and we confirmed again that in undifferentiated pleomorphic sarcomas, which are the closest model to, which are the closest human tumor to the model that we used, methylcolanthrin carcinogenesis, there is strong prognostic significance. So this is the general scheme, that the lectin pathway in sarcomagenesis is triggered by aberrant glycosylation, complement activation. The effector molecule is C3a. There is a regulatory pathway centered on a molecule that we cloned, and this results in tumor promotion on the one hand, and dampening response to checkpoint blockade immunotherapy. So macrophages in tumors are diverse. They are shifted in a pro-tumor, they are set in a pro-tumor mode by signals that come from adaptive immunity, from cytokines, and complement, and this is the most recent work that we have done. So the challenge is translation, and I want to touch upon prevention, interception, pharmacologic targeting, and then I'll go back to where we started from, that's pathology. So I lived at a time when there was only one cytokine associated with inflammation and macrophages, IL-1, so we contributed to the dissection of the diversity of components of the IL-1 system. We identified the type 2 receptor as a decoy, we cloned one of the receptor antagonists, and we cloned this fringe member of the family that we now call IL-1R8, and showed that MYD88 is downstream of TLR. So early on, we asked the obvious question, if macrophages promote cancer, does IL-1 promote cancer? And this is prehistoric data, last century data. And indeed, we found that IL-1 did promote cancer. And this was confirmed by many people. And in particular, I want to emphasize the key work done in Israel by Ronnie Apte and the late Ronnie Apte, because Ronnie did genetic studies with IL-1 receptor knockout mice. Then after 30 years, here is the Canto study, cardiovascular. Here is Pasquale. Cardiovascular, patients at risk of complication from atherosclerosis. In my view, it's a carcinogenesis studies, human carcinogenesis, but I may get back to that. Blocking IL-1 beta, and probably the most striking data, these are 10,000 patients, a 60–70% reduction in the incidence and mortalities from lung cancer. Of course, we have a better understanding of mechanisms that include recruitment and skewing of myeloid cells, production of humoral components of the humoral arm of innate immunity, skewing of, inappropriate skewing of adaptive responses, and more. And of course, this raises the issue of a pharmacoprevention trial or interception trial. There is a group of institutions in Italy called Canal that have a protocol on that. I do hope, of course, all pharmacoprevention studies have failed in cancer, but I think that this remains an open issue, and it should be done. Myeloid cells are a target for pharmacologic intervention in cancer. There are myeloid checkpoints. My count is, and this is the recent review, our count is that there are 13 of them, and for 3 of them there is some noise in the clinic. For instance, Clever-1 and LILRB4. And in general, there is a general, I would feel that in general, these therapeutic strategies targeted on macrophages in tumors have a chance if appropriately combined with strategies targeting T cells, so lymphoid cells. Lymphoid cells, well, we stumbled repeatedly on the interaction of myeloid cells with innate lymphoid cells, and I want to show 3 examples of that because of general significance to this meeting. The first one is this molecule, IL-1R8. We were the first to clone this gene, and we focused on this gene because in the TEER domain, we spotted 2 amino acid substitutions which to us suggested that it should not be a signaling or part of a signaling receptor. And indeed, work done by us and by Shi Shaoli in the United States has shown that this is a negative, as we supposed, is a negative regulator of members of the IL-1 receptor family. It is recruited at signaling complexes of the IL-18 receptor and downregulates signaling. In addition, and we contributed the tools for this work, although this is not really our work. This molecule is part of the receptor complexes that recognizes IL-37, which is a negative regulator, anti-inflammatory cytokines. And when we targeted the gene and when we blocked the gene, and this molecule is very high in NK cells, when we blocked the gene, we unleashed activation of NK cells or innate lymphoid cells and protection against primary carcinogenesis and metastasis in the liver and not elsewhere. And we think, so the tissue context is essential, I think, in general, as we were discussing over coffee. And no phenotype in methylcholanthrin carcinogenesis. And I think that this is related, we think that this is related to the fact that there is plenty of innate lymphoid cells in the liver to start with. And this is more recent work, and the author of this paper, and in collaboration with Lorenzo Moretta, and this is more recent work. They have a strategy to silence the genes in human NK cells, and indeed, silencing of the gene is associated with increased activation, cytokine production, and effector function. The second example has to do with the dark matter in the genome. And this is a story that started many years ago when Fernando Martinez, a fantastic PhD student from Cuba, did the first transcriptional profiling of human monocyte to macrophage and then polarized activation. And among the genes that popped up, and this was supervised by Massimo Locati at the time in my lab, we are still working together on this. And among the genes that popped up, we were interested in MS4A4A because it looked to be restricted to macrophages and nobody knew what it does. And there was a further reason for that, is that it's a tetraspan. The family, the relative is well familiar to all of us, CD20, but the rest of these molecules have unknown function. They belong to the ignorome, the substantial fraction of the genome which encodes protein that we don't know the function, about 15%, I understand. So Irene Mattioli in the lab has worked out the mechanism. It is essential platform that for signaling by innate immunity receptors. Our own work is Dectin-1, but there is more work on TREM2, independent from us. And it's essential for sick activation, signaling upon recognition of fungi, tumor cells, triggering, an NK cell-dependent pathway of resistance to metastasis. But what I think is interesting is that there is a very strong genetic association with neurodegeneration, Alzheimer's disease, which again points to inflammation and this is more recent work that we have done together with Cos Pizzalis at Queen Mary and Marie-Astrid Boutet, and this is unpublished work. Myeloid cell differentiation is associated with a changing repertoire of tetraspan and exposure to, for instance, glucocorticoids is associated with upregulation of a distinct repertoire of these tetraspan molecules. And we have indications, again, together with Costantino, we have indication that this is important for the activity, for regulation of the activity of glucocorticoids in models of autoimmunity. Last example, and I'm really showing this example only to emphasize this. We are doing single-cell analysis. In this particular case, again, sarcomas, and by single-cell analysis, Andrea Ponzetta identified the protecting function of myeloid cells, a tripartite interaction involving neutrophils, macrophages, unconventional, double-negative alpha-beta T cells. And this minute population is complex, and this is single-cell analysis. And the general message is, this is Georges Seurat, pointillisme. The general message is that we are doing single-cell analysis, but pointillism, but we should not miss the Tour Eiffel, I think, and sometimes I have a sense that, and I will get back to that if I have time with some unpublished data. So back to pathology, and we have known for a long time that in general, high macrophage infiltration is associated with bad prognosis. This is generally true, but there are some exceptions, some interesting exceptions. And this is a case in point of recent, or relatively recent work. This, again, liver metastasis from colorectal cancer. Liver metastasis, an aggressive surgeon, Matteo Donadon is an aggressive surgeon. A good aggressive surgeon can take care of about 20-30% cure, 20-30% of the patients. But of course, they don't know who is going to be cured or not. So we looked at macrophages, and then people came to me and said, look, there are macrophages in these tumors in the invasive front that look different, with different morphology. Small guys, big guys, and I happen to know, I have known, all of us have known, the macrophage students have known for ages that these cells, upon exposure to different signals, have different morphology. So what we have done, on the one hand, we have done RNA-seq and single-cell analysis. I'll get back to single-cell analysis. On the other hand, they have done a machine learning approach to have a quantitative algorithm to quantify the small guys and the big guys. And it turns out that the big guys have very strong prognostic significance. And they have a distinct repertoire of genes expressed, including lipid metabolism, complement, we go back to complement, and more. And this has now been confirmed in an independent study done at the National Tumor Institute in Milan, which is now in press, in a surgery journal. So, check my time. So this is so again, single-cell analysis. And we have done, as I said, single-cell analysis of macrophages in tumor, in this particular case, in liver metastases. There are 18 clusters. I don't use the word subpopulations. I use the word clusters. 18 clusters. And I want to emphasize the approach that we have used. Comparison between normal adjacent liver and the invasive front, and selection of the ones that pop up. And then comparison of tumors with fibrosis in the liver and with lipid pathology in the liver. And of course, the relatives, the Kupffer cells, the resident cells. So we came up with a signature with a gene associated with a signature, and I didn't even know what it is. It's GP and MB. And it's expressed by big guys. And then there are small guys, again, the same approach. 3 populations that are, and this is pseudotime, monoclonal pseudotime analysis, probably downstream of differentiation of young monocytes. And again, and these are the small guys, And a marker gene, which in our hands is SERPIN-B2. I should mention that these cells also express SPP1, which Zemin Zhang has used as a marker gene for a subset of the tumor-associated macrophages in Beijing. Is it associated with prognosis? And these are our own case list. The GP and MB positive cells are strongly associated with bad prognosis. And this is something that usually people don't do, and people in pharmacology do, of course, multivariate analysis. And multivariate analysis shows that this is prognostic, independent of other variables, including KRAS mutations and therapy. These are liver metastases, this is TCGA, this is the primary tumor, and again, the same gene popping up. And then you do spatial transcriptomics, and you realize that, and we found that the genes are differentially expressed and that the population are differentially localized. And I can skip this one, go back. So we have done single cell analysis and identified marker genes which are differentially expressed. The big guys, GPNMB1 positive, are located in the tumor at the invasive front. The small guys are located at the invasive front. They are associated with T cells and engage in an intimate relationship with tumor cells and T cells, presumably suppressing T cell function. These cells, the small guys, originate from the blood, we assume, based on pseudotime analysis, and the big guys probably originate from monocytes through a Kupfer-like step of differentiation. Strong, independent prognostic significance, but I think we have to go back to the Tour Eiffel. So what is the meaning of, and this is speculation with some data, I should say. GPNM is a largely forgotten cytokine, which has been shown to skew macrophage function and to suppress adaptive immune responses. And we think that this may underline the strong prognostic significance and biology of these cells. Finally, macrophages are sexy, so sex and macrophages, and we have known for a long time that response to checkpoint blockade immunotherapy is different in males and females. And this is non-small cell lung cancer. Fabio Conforti, who is now in our institution, looked at, in 2,500 patients, at the tumor microenvironment in males and females, the same tumor, non-sex related, and profound differences. And I think we have known, we heard talk about vaccines, that there are differences in immunity between males and females, and there is difference in response to checkpoint blockade immunotherapy. There is difference in macrophage polarization. This may be related, for instance, there are a number of genes that are related to innate immunity that are localized on chromosome X, and escape of X inactivation may be an element. Of course, there are differences in behavior, and there are sex-biased effects. And I think we should have a gender perspective also for non-sex, non-gender related tumors. And the relation between inflammation and cancer, I focus on the tumor microenvironment, but is key at a systemic level for the connection between obesity, lifestyle, and aging. And finally, we heard, for those of you who were here, we had a fantastic talk about COVID-19. And again, in the same review, New England Journal, Cecilia and I summarized the interplay between innate immunity and SARS-CoV-2, and we were involved in that, again emphasizing the key importance of components of the humoral arm of innate immunity, either as biomarkers in this particular case, or novel mechanisms of resistance, and this is the case for MBL. I will stop here. I hope I did not forget to mention the people who have done the work and the collaborators, and this is our own institution, and in our hospital we have a joint effort with the Brera Comedy to put together beauty in a hospital context. So thank you once more. I think we have time for questions.
Thank you, Alberto, for the beautiful and fascinating talk. If you have any questions, there are different microphones and different area of the room, so please, number 3.
Thank you very much for your excellent talk. So I have a quick question for you. So you talked about this tetraspanin in macrophages. So there are 2 questions around this topic. Do other, myeloid cells express this receptor? And is this a druggable, thing?
Yeah.
I mean, are there any pharmacological manipulation going on here? Thanks.
Yeah, well, first of all, there is differential distribution of the MS4A family members. A1 is in B cells, and we are using, it's CD20, so that is druggable. We are using anti-CD20, of course, in diverse conditions from cancer to autoimmunity. Other members of the family are pretty restricted to myeloid cells. For instance, A3 is expressed in the precursors, and people are using to track myeloid cells. Neutrophils, and immature neutrophils are A3 positive, for instance. And in terms of druggability, well, the principle is CD20, and so I assume they are an interesting target, but the only example is CD20.
Thank you.
So I can ask one question. Alberto, you have given a very comprehensive overview of the immune cells' contribution to different kinds of cancers, but you have emphasized that the interplay with the tissue and the environment is fundamental. So which somatic cells you think are fundamental in different scenarios and that we may target also for therapeutic utility?
Yeah, I feel that different tissues, if you take, for instance, the skin, you have a gradient of members of the IL-1 family from the upper skin to down skin. And those are the epithelial cells. It's clearly you have IL-18 and 37, and there is a reciprocal distribution in the epithelial cells in the skin. And then you have tissue-resident cells, of course, in the skin. The liver is rich in myeloid cells. We have in the clinic, we have a problem with, a major problem with bone metastasis. And there is a recent data report in the JEM, General Experimental Medicine, showing how distinct is the interplay between myeloid cells in the bone relative to other organs. And there, of course, you have the point, the general, they play a key role in morphogenesis in the bone. And so I think that the rules of the game are different in different tissues, and we should start taking that into consideration.
Okay, thank you.
Yes. May I ask a question? First of all, a great lecture, as usual, and also great integration of art and science. I wonder if you ever look whether the immune checkpoint inhibitors or ligands actually for the immune checkpoint inhibitors on macrophages, and we can call it maybe T8 as one, Siglec-7, Siglec-9, changes their phenotype or could change their phenotype toward from being pro-tumorogenic to anti-tumorogenic. What is known about?
Yeah, the PD-1 has been suggested to be expressed in macrophages, in tumor-associated macrophages. And it has been suggested that we see expression, but not such a fantastic expression in our hands. And it has been suggested that these anti-PD-1 targeting antibodies may actually regulate the function of tumor-associated macrophages. And as far as Dectin-1 and it skews, for instance, Siglec, one of the Siglec members, 7A, skews myeloid function in a type 2 direction, in pro-tumor direction.
Okay, thank you.
If there is no more question from the audience, Alberto, thank you very much for the contribution.
Yes, it's my pleasure now to invite my co-chair, Professor Pasquale Maffia, to deliver his lecture on targeting inflammation to reduce cardiovascular disease. Professor Maffia is a professor of cardiovascular immunology and is an international lead of the School of Infection and Immunity at the University of Glasgow and co-director of the Aura Cluster of Excellence that links the best African university to also top university in Europe. And last but not least is the new chair of the Immunopharmacology Committee of IUFAR. So the floor is yours.
Thank you, Francesca. So, morning everyone. I will be discussing, I will switch now on cardiovascular disease rather than cancer, and I will give you an overview of what we know in terms of immune response contribution to cardiovascular disorder and if this can be a pharmacological target. So cardiovascular diseases are the major cause of death worldwide. In the UK, they are killing 27% of people. This is according to the British Heart Foundation. The good news is that in the last 40-50 years, we have seen a decline in cardiovascular disease-related deaths, especially due to the introduction of drugs, and that is why pharmacology and pharmacotherapy is important. Statins were introduced around this stage, and clearly they are working. But this decline is coming to a massive cost to the national health system in terms of primary and secondary prevention. And this is not sustainable worldwide. If you look at the global picture, you will see that a pathology that was prototypically considered associated with a westernized lifestyle is today killing more people in Africa and in Asia compared to North America, Europe, and Australia. And I will touch on Africa later on in this talk. The pathology behind the majority of cardiovascular disorder is atherosclerosis, which is a slow and chronic inflammatory disorder leading to plaque formation in large and medium-sized arteries. And of course, you all know the clinical manifestations in terms of myocardial infarction and stroke. The picture of atherosclerosis has been changing constantly in the last 100 years from a disease that was considered mainly driven by hyperlipidemia, and this is still the fact. In fact, hyperlipidemia is the major trigger, to a pathology that now is fully considered to be an immune pathology with immune cells contributing to all the different stages of the pathology. And this changed in the 90s with some pioneering work from Peter Libby and Göran Hansson on the topic. So my group has been contributing to the understanding of immune mechanism to the pathology in the last 15 years. We were the first showing by two-photon microscopy that actually in the mouse model, immune cells, in this case T cells, are infiltrating the vessel wall from the adventitia and the perivascular space rather than from the luminal side. So they are all entering from the back in the vessel. And actually, this accumulation of immune cells, at least in the mouse model, in the very late stage of the pathology, takes form of tertiary lymphoid structure, which are basically ectopic lymph node, fully structured. And we have done a lot of work in collaboration with Andreas Heibenich to demonstrate that actually they contribute to the pathology and they are not an epiphenomenon. These tertiary lymphoid organs that we name artery tertiary lymphoid organs are compartmentalized in terms of T cells and B cells and germinal center. They develop always in correspondence of the plaque, but in the adventitia, in the perivascular space. And again, by two-photon microscopy, we demonstrated, here we are looking at an aorta of atherosclerotic mice. Dendritic cells are or anyway, myeloid cells are in green and T cells are in red. And when you give a cognate antigen for these T cells, you can see that in only few minutes, they are interacting with dendritic cells. The blue is the fibrillar collagen in the adventitia. So we are in the vessel, and as you can see here from the movie, there is the initial step of adaptive immunity. Now, we know that this accumulation of immune cells in the adventitia is driven by a molecule called the lymphotoxin beta receptor, which is actually expressed by somatic cells. In the case of the vasculature, is expressed by immune cells, but in the case of the vasculature, is expressed by smooth muscle cells. And what we did, we created mice that are conditional, that are atherosclerotic, but are conditional knockout for the lymphotoxin beta receptor. And in these mice, there is less accumulation, as you can see from the bright field, of immune cells in the adventitia. And most importantly, the compartment of T and B cells disappear completely. But when you disrupt the formation of this tertiary lymphoid structure, actually, when you look at the pathology here for the one that are not expert in cardiovascular disease, we are staining here with oil red or lipids, so more red, more plaque. You can see that in these animals without tertiary lymphoid structure, there is more pathology in the abdominal part of the aorta, where is the lymphoid structure. This is adding an extra layer of complexity, showing that, of course, immune response can be detrimental and beneficial depending on the context. More recently, the same group of collaborators together with Peter Olofsson, which is here and will be speaking after me, I'm sure will be elaborate more about that. They have shown that actually this infiltration of immune cells in the perivascular space is directly regulated by the brain, and so now we should take into account not only the vasculature and the immune system, but also how the central nervous system is contributing to peripheral inflammation. In the early stage of the pathologies, at least in the mice, we have been studying a lot of immune cells. In this case, we studied the plasmacytoid dendritic cells, which acquire a unique phenotype in the atherosclerotic vessel. They express MHC class II, and they are working as antigen-presenting cells, being able to induce T-cell activation and clonal expansion, while usually these cells are not strong antigen-presenting cells, and they work actually on the interplay between innate and adaptive immunity. This is to demonstrate that the environment is important to shape the activity of the immune cells. This is the first paper published on using CyTOF to look at the diversity of immune cells in the atherosclerotic aorta of mice. This was done in collaboration with Claudia Monaco in Oxford. And as you can see, we have a broad range of different immune cells, lymphoid, myeloid, in the vessel wall. But most importantly, 2 years later, we demonstrated that a very similar picture is present in the naive aorta of the mice. So in the absence of any pathology, you can see a very diverse population of immune cells present in the vessel wall. And actually, some of these cells are naive cells, and they are able to sustain priming and activation and clonal activation of immune cells directly in the vessel wall, almost showing, I know that this is a little bit of a stretch, but that the aorta is a sort of lymphoid organ and can sustain the key steps of immune cell activation. So, where are we with the other cardiovascular disorders? So we do a lot of work in hypertension. The study of immune response in atherosclerosis came first. Hypertension has been following, and the big change in terms of hypertension is this paper that was led by Dave Harrison and Thomas Gudzik in 2007. Thomas is my main collaborator since several years now. And actually, in this paper, they demonstrated that in RAG1 knockout mice, which are deficient of mature T and B cells, you have less hypertension. And actually, this is driven by T cells. And as you can see, since then, a lot has been done in the field. Now we know that there are several immune mechanisms regulating hypertension and also regulating the end organ damage of hypertension in the vasculature in terms of endothelial dysfunction and vascular remodeling, but also in the kidney. We have tried to translate all of this knowledge, especially in hypertension, in the clinical arena, using different kind of approaches. By Mendelian randomization, we have shown that there is a clear correlation between circulating white blood cells and blood pressure. Here, using a multi-omics platform, we have shown that immune mechanisms are key in kidney damage driven by hypertension. And more recently, we have just published on European Heart Journal that actually there is a close interplay also between hypertension and the brain, so the brain is coming back again. And we have identified specific areas of the brain which are affected by hypertension through working through UK Biobank. The picture is similar in other pathology. Today, the big problem in cardiovascular disorder is heart failure, which is affecting an aging population. And again, in heart failure, there is a lot of knowledge in terms of the systemic and the local contribution directly in the heart of immune cells. Heart failure is slightly behind in terms of knowledge of immune response compared to atherosclerosis and hypertension. And here in Glasgow, we have some of the key players in the field. John McMurray was giving a keynote lecture 2 days ago from this stage. I'm working a lot with John Cleland and Pierpaolo Pellicori, and we are trying to address this at the clinical level. So where we are in terms of pharmacological targeting inflammation and immune response in atherosclerosis. The first key trial was published in 2017, is the trial that has been mentioned already by Alberto. This was led by Paul Ridker and Peter Libby. It's the CANTOS trials in which they are treating people with already a myocardial infarction, so this is secondary prevention, with canakinumab, which is a monoclonal antibody targeting IL-1 beta, and they've shown a significant reduction of secondary major cardiovascular events. However, as Alberto said, the drug has not been approved for treating cardiovascular disorder. The most striking results were related to cancer, where actually has been shown that blocking IL-1 beta is beneficial, and Novartis, that was the sponsor of the trial, decided to go ahead in the direction of cancer. However, 2 back-to-back studies were published later on, Colcott and Lodoco-2 trial. In this case, they are using a very similar approach, secondary prevention, but the drug is colchicine. They are both successful. And actually, the important point is that just 10 days ago, basically, the FDA has done the first approval for the use of colchicine. 0.5 milligram is the Lodoco tablet for the secondary prevention of cardiovascular disorder. So we are at the very early stage compared to cancer, but basically now we have the first drug approved for secondary prevention that is targeting immune response. Colchicine is a very pleiotropic drug, is doing several things, but in the trials they've shown that it's mainly affecting the inflammasome and the IL-1 and IL-6 pathway. So what's next? This is just early days. There are several issues that should be addressed. I like to discuss them in my lectures, but of course it's just my point of view and some foods for thoughts. So I think it's important to translate what has been done in atherosclerosis in other cardiovascular disorder, in a sub-analysis done in the CANTOS trials. In blood pressure, canakinumab was doing nothing on blood pressure control, although we can stratify the patients on blood pressure, and the one with the highest blood pressure were the one leading to to a better effect of the treatment in terms of secondary prevention. In a meta-analysis that we published 2 years ago, we have shown that actually in mouse model and in human, only targeting TNF-alpha is leading to reduced hypertension in both human and mice. But still there is a lot to do. In heart failure, there has been no clinical trial targeting inflammation so far. The other big issue that I think we should take into consideration is the global picture. I told you that in Africa, 50% of people are dying of cardiovascular disorder, and this is striking in particular if you consider that the average, the median age in Africa is 19.7 against 44.4 in the UK. So this is the youngest continent on the planet, and despite that, these young people are dying of cardiovascular disorder. Why? Of course, there is genetics, but the environment is extremely important, urbanization, social deprivation, the interplay with infection, which with COVID became relevant also in Europe, but in Africa, consider how many times along the lifespan people in Nigeria are infected by malaria, which is actually interacting with the endothelium. So we are trying to address some of these questions through the Africa-Europe cluster of research excellence on non-communicable disease. This is a large enterprise driven by the European Commission and the African Research University Alliance, which is a sort of Russell group of the top research-intensive African universities. I am co-leading with Tom Gudzik and Paris Cité this large cluster in which we bring together Francophone and Anglophone African countries, and we will try in the next 10 years to address some of these questions if you are able to. What is the real source of vascular inflammation? Of course, there are the classical risk factors, is a fact, but in this paper we have recently shown that, for example, treatment of women with breast cancer with docetaxel was causing vascular dysfunction through actually increasing oxidative stress. And this is something that is almost never taken into account when we are calculating the risk factors in patients. So what is your previous history of treatment and comorbidity? So this is another thing that should be explored more. And pharmacology should do more. In particular, in atherosclerosis, we know that the major immune dysfunction is present in the vessel wall, but not systemically. But the delivery of immunosuppressant directly to the vessel wall is very complicated. I've been working as a lab a lot on the field, in particular in collaboration with the Italian Institute of Technology, but there is no platform in clinic currently used for delivering specific where they are needed immune treatment, and this is something where we should also focus. And finally, I will finish in the last 5 minutes focusing on novel mechanisms that I think should be taken into consideration. The industry has been very conservative, using the same drugs in all the chronic inflammatory disorder, and I really think that we should identify key molecules that are important in specific pathologies. Now, in immune response, inflammatory chemokine receptors are extremely important in regulating immune cell trafficking. They are expressed by the majority of immune cells and myeloid cells. But the targeting in terms of pharmacology of these inflammatory chemokine receptors, which are namely CCR1, CCR2, CCR5, is very complicated. And this complication comes from several reasons, but one of them is redundancy, because a lot of ligands, a lot of chemokines are interacting with these receptors. And we need to, there is the risk that blocking one of the receptors may be useless because the ligands are interacting with the other. So this may be one of the reasons, it's not the only one, why several trials targeting these molecules have failed, while we know at the mechanistic level that they are important in chronic inflammatory disorders. So in terms of atherosclerosis, we are doing a large program of study on these molecules in collaboration with a key collaborator from the University of Glasgow, which is Professor Jerry Graham. These data are actually from Peter Olofsson and the biobank from the Karolinska Institute, where we show that actually there is a significant increase of all of CCR1, 2, and 5 in plaques compared to normal arteries, and also these molecules are increasing in symptomatic versus asymptomatic patients. Similar data we found in the AtherExpress biobank from Gerard Pasterkamp in Utrecht. By single-cell sequencing, we know that the CCR1 is the highest one expressed in the plaque, mainly in TREM2 macrophages. CCR2 is present in monocytes, and CCR5 in the plaque is expressed on T cells. And all of these receptors are actually correlating with what we call the plaque vulnerability index, which takes into consideration several features of plaque instability. So how we are trying to address the redundancy issue? Jerry created novel mice in which the entire locus producing basically inflammatory chemokine receptor, which sits on chromosome 9, has been deleted. So now we have knockout mice that we call taco mice, in which CCR1, 2, 3, and 5 are absent. We have induced the atherosclerosis in these mice, both in male and female. You can see that there is a drastic reduction of plaque formation. No major changes in terms of plaque composition. This is in atherosclerosis an early stage. This is an atherogenesis study. When we normalize the effect on the global knockout mice compared to the different individual knockout mice, we discovered that actually CCR2 knockout has exactly the same effect of the TACO. So this is the first demonstration that actually, at least in this model, there is no redundancy and is all driven by CCR2. So we should target CCR2 to reduce infiltration of immune cells. In the later stages of the pathology, similar data, clear reduction of the plaque, but here we see also reduction of future of plaque instability, like less necrotic core, less calcification. When we look at CyTOF, we see that there is a selective modulation of immune cell infiltration in the aorta in the knockout mice. In particular, we see less inflammatory monocytes and inflammatory macrophages, less conventional dendritic cells of type 2, and more anti-inflammatory macrophages defined by 206. So this is a fine way to selectively modulate myeloid cell trafficking to the vessel wall. T cells are not changing at all. And so we think this may be important, and this is reflected also at the vascular level, where actually we see more smooth muscle cells in the knockout animals, and this is leading to a more stable plaque phenotype. So this is to show that we can identify still a lot of targets that play an important role in the pathology, and probably I think we should change the way of thinking targeting immune response in a secondary prevention, but maybe we should actually aim to really immunosuppress people at very high risk for a short time frame in a controlled manner, reset vascular inflammation, and then leave them on standard care. I mean, this is all for me today. I would like to thank, of course, all the people from the lab and all of the main collaborators and fundings. Thank you.
Thank you. The lecture is open for questions from the public. I see Alberto, please.
That's great. Is it working? Yes. Very, very nice. What about localization in the plaque of the population as driven by the different chemokines in the last part? Is localization important?
Yeah, so I don't have data in terms of human, because in human these are endoarterectomy samples, so basically they are plaques that have been dissected and then digested, so we don't know exactly where the data are coming from. We have not done any spatial transcriptomics In the mouse, we know that they are all infiltrating from the perivascular space. So the majority of immune cells are present in the perivascular adipose tissue and in the adventitia. The majority of the lymphoid cells are in the perivascular tissue and in the adventitia. The myeloid cells, in particular macrophages, are more in the plaque. But the more the plaque develops, the more inflammation is growing in the perivascular space. In fact, Haralambos Antoniades, which is a friend and colleague from Oxford, has now patented and is having a lot of success, a way of imaging with CT scan perivascular inflammation as a biomarker to predict cardiovascular disorder. So it's the perivascular space which is key.
Yes?
I'm Sachikin from Southampton. I'm very, very new to the field, so treat my question as that of a student. Is the inflammatory response a cause or an effect to the initial atherosclerosis by the lipids and so on? And then I think you answered my question, but what you've just said, is there a stage in the inflammatory process where it gets burnt out and you get the fibrosis and the blocking, and the inflammatory response perivascular doesn't affect the patency of the artery? Or is that the inflammation surrounding it, if it's suppressed, it has a meaningful clinical effect in increasing the patency of the already blocked artery?
So, remind me the first question.
The first question, is it a cause or an effect, the inflammation?
Yeah, so of course we don't know. So it's a fact that hyperlipidemia is the trigger, but it's a fact that I've shown to you there are immune cells in the vasculature at any stage, even in the absence of the pathology. As soon as you give a trigger, for example, you induce hypertension in mice, in 3 days you see a massive infiltration of immune cells. So there is an initial insult, but then the infiltration of immune cells is very significant and they take control of the tissue damage. In terms of your second question, again, I don't have an answer, but I think you are raising a very important point, which is targeting immune response is complex and is depending on the tissue, is depending on the timing, so when we should target and why. So these are all unanswered questions. We need to stratify, first of all, patients better. The one that responded better to canakinumab, for example, were the one with high C-reactive protein in a very similar trial done afterwards, when the patients were not selected to be with inflammatory residual risk. There was no effect of anti-inflammatory treatment. And we should identify better when targeting inflammation would be detrimental or beneficial, because of course the immune system is very plastic and is doing different things in different contexts.
Can I ask a third question?
Sure.
Is there any speculation whether the beneficial effect of aspirin is due to its antiplatelet function or its anti-inflammatory function?
No, aspirin is mainly working through platelets, and there is also a lot of controversy on the use of asthma as antiplatelet. The anti-inflammatory effect of non-steroidal anti-inflammatory drugs is actually shown to be detrimental in experimental models.
Thank you.
If I might add a point to this answer, I think Mauro Peretti is in the audience. I cannot see. Yes, here he is. To add a point, and after I have two questions, but maybe later on, aspirin, it has been shown to cause the formation of lipoxin A4, as well as statins. And this, why I look for Mauro Peretti here, because they are pro-resolving lipid mediators, and therefore they would downregulate inflammation that we have. So it can be that at a given point, as in any inflammatory response, inflammation is protective, but if it becomes chronic, it's not protective. And you see that the body itself, I mean, aspirin is inducing the formation of lipoxin. Statins are inducing the formation of lipoxins. However, we also produce these stopping signals of the inflammation. This is my opinion. If Mauro would like to say, add something, I would love it. Yes? Later? No, now, please. So you can, please.
Thank you, Francesca. No, I'm not sure. Well, I think you're perfectly right. The complexity of aspirin is more than simply inhibiting COX, because aspirin, as we know, produces a covalent inhibition of the active site of the cyclooxygenase. There is evidence that once the active site of the COX1 and COX2 is acetylated, then arachidonic acid synthesis can produce aspirin-triggered lipoxin A4, which are modulatory, pro-resolving, increased macrophage phagocytosis, might switch the macrophage. There is evidence, I think, by Ira Tabas, by and large, in the context of atherosclerosis, which is the topic of the excellent lecture of Pasquale. Ira Tabas has published quite a streak of studies whereby pro-resolving mediators and molecules can modulate atherosclerosis and attenuate the inflammatory response. So thank you for calling me in. I'm enjoying the symposium. I thought to be Thanks.
Thank you, Mauro. And can I ask you a question, or we are out of time? Because you are being my co-chair, you're...
Yeah, I switch off the timing, but anyway, you
can
answer one quick question and then we go ahead.
Yes, I was wondering, being my loved cells, what the muscles do in the plaque. So you said, and it's very important, your lecture also for that we have to be aware that there are resident immune cells or innate immunity cells in the aorta. So what about mast cells?
Yeah, so mast cells actually, in terms of location also, and what Alberto was asking, they sit exactly at the shoulder of the plaque, which is where the plaque breaks. And there was a very nice study done by Gerard Pasterkamp a few years ago, demonstrating actually that they contribute to the instability of the plaque in human. There is a lot of work also done by Shi and Peter Libby in animal models, demonstrating that in animals knockout for mast cells, there is less plaque formation. So they are involved and they play an important role.
Okay, now we can sleep well. So I think we have to move to our next speaker. Thank you again. And you would like to introduce him?
Okay, so it's me again. The next speaker is Lucy Macdonald from the University of Glasgow. Lucy has been doing some seminal work using single-cell sequencing for looking at myeloid cells in synovial tissue in rheumatoid arthritis, and she's a talented early career researcher that will present the work. Thank you, Lucy.
So good morning, everyone. Thanks for the introduction, Pasquale, and the invitation to speak to you all. So as Pasquale said, I'm a postdoc in Mariola Karauska-Stolarska's lab at the University of Glasgow's School of Infection and Immunity. And today I'm going to be presenting our work where we have identified distinct synovial tissue macrophage subsets which regulate inflammation and resolution in rheumatoid arthritis. So rheumatoid arthritis is a systemic autoimmune disease that is characterized by chronic inflammation of the joint synovium, and this results in persistent pain, swelling, and stiffness. And in recent years, disease-modifying anti-rheumatic drugs have revolutionized therapy for RA, and this is through their targeting of the inflammatory pathways and the ability to prevent disease progression. Perhaps the most famous group of biologics for treatment of RA are the TNF inhibitors, and these drugs target the macrophage pro-inflammatory cytokine tumour necrosis factor. And so macrophages, as we've discussed earlier on, are the effectors cells of the innate immunity, and the success of anti-TNF therapy highlights the role that macrophages play in rheumatoid arthritis. And you can see in this schematic here that macrophages dominate the synovium in RA disease, and their prominence increases with disease severity. However, we know that macrophages also exist in the healthy synovium, and they form a protective barrier through interactions with the lining layer fibroblasts to form the interface between the joint cavity where the synovial fluid exists and the sublining tissue. In addition, we know that macrophages also persist in remission of rheumatoid arthritis, whilst their numbers decrease. And the result of recent advancements in therapy for rheumatoid arthritis has made remission an achievable goal for more patients than ever before. However, 50% of patients still flare. And so this highlights the clinical needs for targeting areas of resolution rather than just the inflammatory pathways. So this brings me to the first aim of my project, and that is to uncover the heterogeneity of synovial tissue macrophages and joint health, inflammation, and resolution of rheumatoid arthritis. So previous work by our group had applied flow cytometry to characterise macrophages based on their expression of Merck and CD206 or Mannose receptor. So Merck is a member of the TAM receptor kinase family, and its function is to facilitate uptake of apoptotic cells through the binding of Gas6 or PROS1, which is the bridging ligand. And so our group previously found that there's two subpopulations of synovial tissue macrophages, the Merck CD206 double positive and the Merck CD206 double negative. And what we find is that in healthy tissue and in patients who are in sustained clinical remission, the double positive population is the most dominant. However, in naive or resistant rheumatoid arthritis, we see infiltration of these Merck CD206 negative cells. And we have tested the significance of this clinically by looking at the effect of the proportion of these subpopulations and the outcome of flare and treatment discontinuation in patients with remission RA. So if you have a proportion of less than 47.5% Merck CD206 double positive, or a ratio of less than 2.5 of Merck CD206 double positive over double negative, then you're more likely to flare upon treatment withdrawal. However, despite this characterization using these two markers, the true phenotypic spectrum of synovial tissue macrophages still is unclear, particularly the difference between the macrophages that we see that exist in healthy tissue to those which persist in remission. And so we perform single-cell RNA sequencing of assorted CD64, CD11b, HLA-DR positive population from the synovium of 27 donors across healthy tissue, undifferentiated polyarthritis, naive and treatment-resistant arthritis, and also patients who are in sustained clinical remission. And we identified 9 phenotypes of macrophage. And so the first, or perhaps the first finding that we found from the single-cell RNA-seq analysis is that CD48 can represent a positive marker of the Merck negative CD206 negative macrophages. As you can see, CD48 is expressed on the cells which are Merck negative, and we know that this population increases in active RA. This Merck negative subpopulation, which we found to be CD48 positive, consists of 3 subclusters, or 3 phenotypes, we could say. The first of those includes the S100A12 population. So this population closely resembles CD14 monocytes, and we think represents cells which have recently infiltrated the synovium, and they have highest expression of CD48, but also high expression of a number of alarmins. And then additionally, we have the SPP1 positive population. So this population has the highest potential for pro-inflammatory activation, and also has a strong glycolysis signature, as well as production of SPP1 or osteopontin. And then we also have the colectin A positive macrophages, which closely resemble dendritic cells and have high HLA expression. On the other hand, we have the Merck positive clusters, which we see dominate in healthy tissue and in remission of RA. And these are composed of two major subclusters, and this includes the TREM2 macrophages. So these are the macrophages which dominate the healthy tissue, and we see that they exist in the lining layer of the synovium at the interface between the synovial fluid. And then also we have the live-1 macrophages, and this is the population that is Merck positive and is most prominent in remission RA synovium, and they exist both in the lining but also around the vasculature. And other literature has previously shown that live-1 macrophages can actually be involved in vascular control of inflammation. And so with these results, this takes me to the next aim of my project, and that is to dissect the function of synovial tissue macrophage clusters and the initiation and resolution in disease. So we know which populations exist thanks to single-cell RNA-seq, but what are these populations actually doing within the tissue? So previous work by our lab looked at the expression of various mediators by Merck positive and negative subclass populations. And so we sorted Merck positive from active RA in remission, as well as Merck negative cells from active RA, and we stimulated them with either LPS or Gas6, which is the ligand for Merck decay, and then performed an ELISA to look at the kind of things that they're secreting. And what we found is that active RA Merck negative cells, upon stimulation, have the highest expression of pro-inflammatory cytokines, such as IL-6, TNF, and CCL2. Alternatively, the Merck positive cells from remission have the highest expression of resolvin mediators, such as resolvin D1. And so, as a result of the single-cell RNA-seq, we found that the SPP1 cluster forms a major part of the Merck negative cells that increase in active RA. And so we were interested in the function of SPP1. So we took peripheral blood leukocytes and we stimulated them with SPP1 at 2 different concentrations and performed single-cell RNA sequencing with the BD Rhapsody platform, which has an immune response gene panel. And we found neutrophils, monocytes, T cells, B cells, plasma cells. And the main finding was that high concentration of SPP1 promotes survival of neutrophils, particularly a phenotype of neutrophils which we refer to as dysfunctional neutrophils. So they're marked by high expression of PDL1, but the rest of the genes in their signature include pro-inflammatory cytokines such as TNF, IL-1 beta, CCL3, CCL4. And in addition to this, we also see a difference in the signature of the CD14 monocytes. SPP1 also drives their pro-inflammatory activation. So we were interested in the function of the Merck negative and Merck positive clusters in the control of the stromal compartment of the synovium. And so we sorted these major subpopulations from active RA and remission RA donors and co-cultured them with active and remission fibroblasts. And then after 48 hours co-culture, performed targeted single-cell RNA sequencing. And what we found is that the Merck negative populations from active RA induces pro-inflammatory activation of stromal cells from the synovium, whereas Merck positive from remission donors induce the stromal repair response. And so we became more interested in the function of Merck specifically. And so here I'm presenting work from my colleague Lucy, who is investigating the function of Merck in mouse synovial macrophages. And so with this model, the injection of tamoxifen removes the expression of Merck from the TREM2 CX3 CR positive lining layer macrophages in the synovium. And then we induce the serum transfer model of arthritis. And whilst we don't see much difference in the inflammatory phase of arthritis, the most significant change is at day 9. Serum transfer arthritis is a self-resolving model. However, with the Merck deficiency, we find that there's a significantly higher increase of the number of leukocytes that are present in the synovium at day 9. And this is associated with a reduction in the TREM2 CX3 CR1 positive macrophages, which typically form the lining layer, and also a reduction in the population of lining layer PRG4 positive fibroblasts that associate with the macrophages. And so this led us to investigate the niche signals and transcription factors that could be driving Merck TREM2 synovial tissue macrophage development and function. And so using tools such as Dorothea and Viper, which perform transcription factor regulon activity analysis, we can look at gene set expression that are regulated by known transcription factors within our single-cell RNA-seq data and predict which transcription factors are active within each of the clusters. And so investigation of the TREM2 Merck-positive macrophage phenotype, we identified LXR alpha, which is expressed by the gene NR1H3, to be a key transcription factor that could potentially be controlling the TREM2 macrophage phenotype. And future work to confirm this will involve analysis of single-cell attack-seq data to look at the open chromatin within these macrophages. However, we looked at validating the function of LXR alpha in the synovial tissue macrophages. And so we took a model of synovial organoids whereby we combine macrophage precursors with endothelial cells and primary RA synovial fibroblasts to create synovial organoids, which we then culture for 15 days. And within the media for the synovial organoids, we either have a vehicle control or we have LXR alpha inhibitor, 22 hydroxycholesterol. And then after 15 days, we digest the synovial organoid and perform single-cell RNA sequencing. And when we look at the macrophages which are present within the synovial organoid, we can identify a Merck TREM2 lining layer-like macrophage, and also this pro-inflammatory TNF-alpha positive cluster. And what we find is that compared to the control, LXR alpha inhibitor leads to a decrease in the number of Merck-positive TREM2 positive cells within the organoids. And alternatively, we have an increase in the number of pro-inflammatory TNF ICAM1 positive cells. When we look at the signature of these cells within the organoids, we also see a reduction in expression of TREM2 and an increase in expression of TNF-alpha in those organoids that have included an LXR alpha inhibitor. Alternatively, we've also looked at LXR alpha knockout mice models, and this is preliminary data, but what we've seen from single-cell sequencing of the synovial macrophages from these mice is that we have an altered phenotype of the lining layer macrophages, which express TREM2 and CX3CR1. So we have a reduction in the homeostatic molecule VSIG4, which is a marker of the lining layer macrophages and is similar to PD-1, and it can have an involvement in preventing T-cell activation as well as inhibiting the alternative complement pathway. So the LXR alpha knockout leads to a reduction in VSIG4 as well as a reduction in the expression of the glucocorticoid receptor, whereas on the other hand we have an increase in the production of CCL2. Additionally, we find that LXR alpha knockout leads to a reduction in the proportion of the aquaporin-1 TREM2 positive lining layer macrophage precursor. So this data is unpublished and has shown the role of LXR alpha in driving the phenotype of TREM2 macrophages in the synovium. However, clinically, targeting LXR alpha would be challenging because previous use of LXR alpha agonists have failed to go past phase 1 clinical trials due to the off-target effects involved in lipid metabolism. And so we were interested in identifying niche signals which are upstream of LXR alpha expression, which could drive the TREM2 macrophage phenotype. And so one of the things that we did was to take single-cell RNA sequencing data from different tissues across different disease states. So here on the left, you can see that we have data from the heart, lung, skin, and synovium. And we find that the PRG4 positive fibroblasts are unique to the synovium. And when we use cell-to-cell interaction tools to predict things that PRG4 fibroblasts could be secreting, that could be regulating TREM2 macrophage phenotype, we identify various candidate molecule mediators such as PRG4, HAS1, which is the enzyme which secretes hyaluronic acid, which is another component of the synovial fluid, CLU, which is clusterin, and APP, which is the amyloid peptide precursor. And when we look at expression between these different tissues, we find that all of these mediators are increased in the joint. Additionally, we find that these PRG4 lining fibroblasts have increased expression of Gas6, which is a ligand for MERTK, in remission RA patients compared to active disease. And so again, we use another tool known as NicheNet, which can predict the mediators responsible for driving your cluster of interest phenotype. And so again, we look at the interaction between the PRG4 fibroblasts and TREM2 macrophage, and we find that APP and ApoE directly influence expression of TREM2 on these cells. And there's been quite a lot of data looking at the interaction of APP and TREM2 in Alzheimer's disease and in microglia, and it's previously been reported that TREM2 combined APP, CLU, and ApoE, and this facilitates the uptake of not only amyloid beta, but also apoptotic cells, such as neutrophils. So in conclusion, we are aiming to reinstate the homeostatic functions of tissue-resident synovial tissue macrophages, which are TREM2 MERTK positive, in order to reinstate synovitis and sustain remission in rheumatoid arthritis. And we have identified niche signals that are important for driving TREM2 macrophage phenotype, such as APP, ApoE, and CLU, as well as signals which are important for regulating their niche-specific functions, such as the barrier function of the lining layer macrophages. And we have identified a potential transcription factor which could be responsible for driving this phenotype, and that is LXR alpha. And so our future work is looking at spatial transcriptomic data. So you can see on the left we have a tissue from active RA and a tissue from remission. And this is just initial analysis of cosmic data, but this will allow us to dissect the interactions of the macrophages within the synovium and how this influences their function in initiation and resolution of disease. So I would just like to say thank you to our collaborators, and for the opportunity to present to you all, and thank you for listening. I'm happy to take any questions.
Thank you, Lucy. Very nice data and very comprehensive, spanning from human to mice, and a lot of food for thought. Is there any question from the audience?
Alberto.
Okay, Alberto, number 2.
A lot of food for thought.
Thank you.
The origin which you hinted at.
Yeah.
Do you have a sense of in the human versus mouse, which is a different, I think, different game in adults?
Yeah, so we've done a lot of work looking at the trajectory of these macrophages in the synovium. And I think a lot of the mouse data suggests that they're embryonic derived, but I guess the mice that are kind of assessed in these are young mice. You know, they're not mice that are of age of rheumatoid arthritis.
My age.
Yeah. And actually we have a single cell data set which is bone marrow transplant donors. So we have the synovium from patients that have had a bone marrow transplant, and using SNP calling we can identify which cells were from the donor and which are from the recipient. Yeah. And from that analysis we find that all of the macrophages in this, or the majority of the macrophages, even the TREM2 macrophages which exist in the healthy tissue, are monocyte derived. We've done other experiments where we have the organoids and we use monocyte precursors compared with induced pluripotent stem cell-derived precursors, and they can both generate this kind of TREM2 MERTK positive phenotype. It's just that the stem cell-derived precursors do a better job of it, so they have higher expression of TREM2 in the kind of regulatory molecules. So certainly you can get the trajectory from both embryonic and peripheral blood.
Number 3.
Thanks, Lucy. My name's Groom Kai from the University of Melbourne.
Hi.
I was just interested in your organoid model, which was quite intriguing. Do they form sort of three-dimensional structures, and is there any difference in the structure depending on the fibroblasts you put in, etc.?
So the organoids are made of the the macrophage precursors, HUVEC endothelial cells, and then primary synovial fibroblasts. And we find, well, actually, it was previously published, I think Kevin Way's the primary author, and they found when they don't include the HUVECs, they see a difference in the fibroblasts, so they don't have establishment of the lining layer when the the endothelial cells aren't present. We have been looking at trying to alter the different subsets of fibroblasts that we include in the organoids, but it's been pretty difficult because sorting the fibroblasts to then put them into the organoids, they've just not been happy, and it's just been, so that's something that we're working on. But certainly we've been looking at the differences in composition of the different subsets from healthy versus active RA versus remission organoids. And it's nice because we can, using a comparison of single cell RNA sequencing from the synovium and from the organoids, we can find the majority of the populations that are present in the human tissue. So it really is a good model and alternative to.
But Lucy, are you using any scaffold, or is it just a co-culture?
No, so it's literally it's just the cells, and they go into the matrigel, and so the matrigel doesn't contain. I can't remember exactly what's in it, but it doesn't contain the normal collagens that you find in the synovium. But once they go in, the cells are into the matrigel, they start producing collagen 1, collagen 3, and you see establishment of this kind of lubricant-producing lining layer. But yeah, it's just matrigel.
Okay, fantastic, thanks.
Yeah,
Francesca.
yes, thank you for your lecture. You describe this novel phenotype of the macrophages in the active synovium with having high CD48.
Yeah.
And we know that the high affinity ligand of CD48 is CD244, 2B4, and this ligand is expressed by many immune cells. Have you an idea of which interactions the high CD48 macrophages cellular interaction are having? What are the partners of the active?
So I hadn't heard of the interaction with CD244. I know that CD48 can interact with CD2.
Also, but in humans, the highest affinity receptor is 2B4, the CD244.
We've not looked at anything like that ourselves, but I know through interaction with CD2, it leads to activation of T cells, so I would assume that it would be some involvement with T cells, but it is quite widely expressed, the CD48, by all the macrophages, so that's why we've not specifically focused on that. But yeah, it's an interesting consideration.
Okay, thank you.
Okay, perfect. Lucy, thank you very much for
Thank
the
you.
very nice talk.
Yes, since we think that the presenter of the oral communication is not present, his name is Gregory Robinson. Is he here? No, he didn't make his way from Ontario. Okay, so I'm happy to invite Professor Peter Olofsson, who is here. I saw you before. He's a professor at the Karolinska Institute of Stockholm, and is the head of the division of cardiovascular inflammation and medicine, and is the founder of the Center for Bioelectronic. It is an integrative system to study diseases. And his lecture will focus now on the neural regulation of inflammation. So we are really trying to cover all the aspects. The floor is yours.
Good morning, and thank you so much for that nice introduction and for inviting me to be here at this exciting meeting. Yeah, so we've heard all morning how important inflammation and its regulation is. And now we're going to turn to a system that, at least in my perspective, not seldom is overlooked when it comes to regulation of this particular system, which is the immune system, which is our nervous system. Now, we know since over 100 years, as described by Charles Sherrington, that there are neural reflexes that regulate organ function. He got the Nobel Prize in the 1930s for describing that simple neural reflexes have at least three components: an initiating organ, a connecting arc, and a target organ. And almost 100 years later, by work by Linda Watkins, Akira Nijima, Ludmila Borovikova, and Kevin Tracy, we know that also this is true in the immune system. There are sensory nerves that respond to cytokines, STAMPs, and PAMPs, and signal through the nervous system, and there are motor nerves that regulate immune cell function. And the first discoveries in this field were made concerning the vagus nerve, which innervates a lot of key inner organs. And therefore, I think a lot of the knowledge that we have today, or the best knowledge we have today on this subject, is about the vagus nerve. Well, on inflammation, I think we still have a lot to learn. This is Emily Brick, who was a PhD student in my lab a few years ago, and she was interested in this fact that people that undergo, that are subjected to health care, in this case, intensive care, for more than 48 hours, they often don't really recover to the state they had before the disease and the treatment. But it's not really known why this is. There is some notion of increased inflammation, but in this cohort, after discharge, the normal usual suspects like TNF, IL-1, IL-6 are not elevated. But she measured the alarmine HMGB1 over 6 months after discharge, and she found that there was a significant increase, even half a year after the patients were discharged from hospital and at home. And she found an association with the cognitive impairment in these patients. Another example of long-term persisting, surprising inflammation is in cancer therapy, where people years after radiotherapy have elevated markers of inflammation in their vasculature. So we were fortunate to work with Martin Halle, who is a plastic surgeon, and when he did reconstructive surgery after this treatment, he was able to take arteries from healthy arteries from these patients and arteries from the diseased region, from the irradiated region, and we found increased inflammation markers in these tissues. And also, as we've heard this morning from several of the speakers, there is now ample evidence that treating excessive inflammation, reducing things like TNF, other cytokines, is beneficial for treatment of disease. So it's certainly worth figuring out how these molecules are regulated and how we can potentially address this better in treatment of disease. So, as I said, the vagus nerve has been studied for about 20 years now in terms of regulation of cytokines and inflammation. And this is Laura Tarnowski, who was a postdoc in my lab at the time. She did this very simple experiment where she subjected rats, or mice, it looks the same, subjected rats to 1 minute, very short time in my opinion, 1 minute of electrical cervical vagus nerve stimulation on this left side. And then she waited a few hours or even a day or 2 days before she injected endotoxin. And what she found was that even after this very brief activation of the vagus nerve, there was a persistent reduction of the TNF response to endotoxemia for 2 days, which I think is a fascinating observation that we don't really fully understand. We have been studying this vagus nerve regulation of cytokines for more than a decade, and we do understand some things about it now. So we know that signals in the motor vagus reach the celiac ganglion, where the adrenergic splenic nerve arises, and this splenic nerve releases norepinephrine in the spleen, which interacts with a subset of T cells that have the capacity to biosynthesize acetylcholine. They release acetylcholine, which acts on alpha-7 nicotinic acetylcholine receptors on innate immune cells and regulates cytokine release from these populations. And on the other side, since it's a reflex, on the sensory side, there are sensory vagus branches, neurons that sense cytokines and PAMs and DAMs. And this system now acts as a reflex. So we were particularly intrigued by these acetylcholine releasing T cells that we found are capable of relaying neural signals. The reason this question arose was because I said, there is an adrenergic nerve that reaches the spleen, and we knew there were cholinergic receptors responsible for regulating innate immune cell cytokine release. So there had to be some kind of connector. And we've found this acetylcholine producing T cell. And that solved the connection problem in spleen. But we thought, are there other organs where there are cholinergic receptors with important functions but very little cholinergic innervation? And one such organ is the vasculature. So another Nobel Prize in 1998 was the discovery of the endothelium-derived relaxing factor. And as part of that prize, it was described that acetylcholine activates muscarinic receptors on endothelial cells, which leads to vascular relaxation. But where does this acetylcholine come from? So we thought, perhaps these T cells might contribute. So we created mice that lack the ability of releasing acetylcholine from T cells. And lo and behold, they had higher blood pressure. And in a project in collaboration with TAC Max Lab in Toronto, we found that this even was important in inflammation and antiviral defense, because without these acetylcholine-releasing T cells, there was not optimal tissue entry of antigen-specific T cells, and there was a lack of the capacity to defend from viruses. Now, these data were all from mouse. So Laura and Vladimir in the lab were interested. So are these cells, do they exist in humans? And yes, they found a way to induce chat, choline acetyltransferase, in cells from human blood. And these cells then release acetylcholine and they regulate, at least in ex vivo models, vascular tone. Interestingly also, supernatants from T cells, not surprisingly, if you expose endothelial cells to supernatants from activated T cells, there's an increase in NF-kappa B activity. But if you take the supernatants from cells that express a lot of choline acetyltransferase and release acetylcholine, the effect is opposite, a reduction in NF-kappa B, which is completely blocked by addition of blockers of cholinergic signaling, atropine and mecamylamide. And interesting, this has effects also on molecules associated with cell-cell interactions So, for example, V-cadherin or zonocludins 1, which is also a cholinergic effect, and also the uptake of dextran into endothelial cells is regulated by in this way. So, which cells in which T cells can have the capacity to make acetylcholine? In a collaboration with Stefan Jovinger, we got access to patients scheduled for extracorporeal membrane oxygenation. These patients are very sick, very highly active inflammation. And what we found was that in these clusters of different T cells, virtually all clusters had a capacity or had examples of chat expression. So it appears that lots of subsets or clusters of human T cells have the capacity to express cholinesterase. In a multivariate analysis, in this small cohort of less than 40 patients, we found that having high relative expression of chat was associated with higher survival than low in a continuous analysis. Should definitely be taken with a big grain of salt because the cohort was very small, but I think it's interesting. So we also heard this morning that the resolution of inflammation is key. Inflammation is usually good. It helps us defend from microbes and helps with tissue repair, but it also has to ultimately resolve. So April Caravaca, a PhD student in the lab at the time, she was interested in this. And she did a simple experiment where she activated the vagus nerve electrically in the cervical region. And then she used the established method to measure resolution of inflammation established by Charlie Sarhan at Harvard, which is neutrophil infiltration and clearance from tissue. And comparing here, and the standard measurement is how long time does it take for 50% of the neutrophils to clear. And in this case, there is a difference from 16 hours to 10 hours. April also developed an inflammation decay index to measure actually how quickly this happens. And she found that activation of the vagus nerve reduced resolution time. Interestingly, part of the explanation is in lipid mediator synthesis, because a lipidomics analysis showed that the ratio between the prostaglandins pro-inflammatory lipid mediators and specialized pro-resolving mediators that we heard about earlier today also was changed by activation of the vagus nerve in a direction that was more pro-resolving. More in detail, there was a significant increase in a few of distinct metabolomes here, DHA, DPA, with increase in resolvins, protectin, merisins, etc. And looking at the interactome, lipid metabolic interactome, we suspected that this 12-15 lipoxygenase enzyme was involved, and mice that lack this enzyme also have an attenuated response to nerve activation in this context. And mice that lack the alpha-7 nicotinic acetylcholine receptor, they fail to respond completely. So this is a cholinergic effect and partly dependent on the ALOX15 enzyme. So April was also interested in different, if this could have effects on different diseases, and this is an example of where she did vagus nerve stimulation in a model of inflammatory bowel disease. And this is probably not a representative example, but a very strong example where the animal that was subjected to the vagus nerve stimulation had a very reduced number of lesions. She did this also in splenectomized animals and found that this is a spleen-independent effect. Which is important because to date, or at least for a long time, the focus was on the inflammatory reflex to the spleen. But her data here shows that there are also other reflexes or other pathways that regulate inflammation. Also in this case, a short activation of the vagus nerve led to a reduction of this response in the intestine for several days. Now, Osman Abend, he came to the lab. He had been working on liver for his entire life. And he did a similar, very simple experiment where he stimulated the vagus nerve in mice, or he cut the vagus nerve, and then he looked at liver samples. And to make a long story short, one of the key molecules, or at least one involved in NAFLD development, PNPLA3, was strongly regulated by signals in the vagus nerve, and so were some inflammatory molecules and some clock genes. Just indicating that these nerve signals have important effects on different aspects of pathophysiology and in a multitude of organ systems, and we don't understand this yet, and we definitely have to study this more. So Pasquale mentioned this study on neuroregulation of atherosclerosis development, and I mean it's known since forever that all vessels are innervated, and the adventitia is innervated in blood vessels, and the atherosclerotic plaque here builds inside the vessel. And looking at how this region looks microscopically in a healthy mouse, there are very few nerves in the adventitia, but as atherosclerosis develops, there is nerve sprouting and increased presence of nerves. And in this project led by Saroj Mohanta and Andreas Habenicht from Munich, we collaborated to find that the vessel is actually innervated by several neural pathways, and perturbing these signals by removing the celiac ganglion changes the course of atherosclerosis. Now, looking closely at this region in the ATLO, we also found that these chat-positive cholinesterase expressing T cells are present here. And it is possible, since these are adrenergic nerves, that they also here relay neural signals, converting adrenergic to cholinergic signals, but that is not yet known. So I have discussed the splenic inflammatory reflex, the vascular inflammatory reflex. Maybe there is a hepatic inflammatory reflex, something that we're working on understanding, and there is this direct vagus innervation of the intestine. So how will we now figure this out? Well, there has been a lack of tools to really study this in detail, because interacting with the peripheral nervous system in small mice or in any animal actually is difficult. So we collaborated with Eric Głowatsky, who is an innovative, very, very smart engineer, and he developed these basically solar cells that can be wrapped around nerves. So with these solar cell-like 4-micron thick devices, they can be wrapped around any peripheral nerve, for example, here in the cervical vagus nerve, and then the skin can be closed, and the laser, long wavelength laser can be used to activate this, just showing here that this activation of the vagus nerve reduces heart rate. So it works. So now we envision that we can use this tool to map these different neural conduits to the liver, to the spleen, to the intestine, and you know, just map this, because that hasn't been done, because it hasn't been possible really because of lack of technology. Also in combination with genetic models, I think this might be a very powerful approach. That's what we hope. And as we learn more about this, we of course would like to take it a step further and integrate this as more of a treatment, personalized treatment device. So we worked here with Simone Fabiano's lab, and they developed this artificial neuron. And the interesting thing with this is that this is a biorealistic neuron that behaves like physiological neurons and can respond to activity in a sensor here. So this sensor can be basically anything. So we connected it to the vagus nerve, and we found that changing the electrolytes here reduced heart rate, increased spiking, etc. And the idea here is that this could be, for example, a sensor for TNF, for glucose, for what have you, for whatever function you want to regulate. And we think this is very exciting. We're continuing this project together with them. So how will we ever translate this to medicine? Because peripheral nerves are inside the body, and up until today, they require surgery by a neurosurgeon to connect electrodes and to activate or deactivate them. There are a number of clinical trials ongoing now, for example, by Setpoint Medical and Galvani Bioelectronics, which is, I think, GSK and Google together, trying to treat inflammation using electrical signals. But there is a high barrier to really get a neurosurgeon and subject people to surgery. So Adam Williamson and Eric Gowatsky, together with us, built upon an invention by Ed Boyden at MIT, which is, to make a long story short, this is published already, so if multiple high-frequency electrical waves are combined, really they don't have any effect on tissue. But if you combine them in a specific way, you can create envelopes, which then can, if you have multiple electrodes, they can really increase focality, and you can get a very precise stimulation, they tell me down to 1 micron, which then potentially can precisely activate nerves. So we made this grid with 20 electrode pairs in a mouse and made the mouse kick. So it works, because we can move the electrical field from the skin and have the intensity only lowered down. So Adam now brought this to human studies using this crude array of electrodes, but showing that this can be used in humans. It can then reduce heart rate, and it simply works, which means that this technology now would enable us to, as we discover more and more neural circuits that regulate different types of physiology, that we can access them without a neurosurgeon and facilitate clinical translation, facilitate our understanding of how these neural circuits work, and simply move the field forward. So therefore, now our focus is to molecularly and cellularly map the neural conduits that regulate inflammation and immunity, and use the novel technologies we have to control nerves, and find ways using genetic models to do this with higher precision to understand the mechanisms, and hopefully translate this into future therapy. Lots of people have contributed to these studies that we're doing. This is my current lab, and we are very happy for loads of fantastic collaborators and great funding. Thank you.
Any question from the audience? If not, I start with my questions, or you would like?
You can start.
You're my yes, I am very interested in the fact, in all your lecture actually, but in the fact that you found that T cells produce acetylcholine. Did you try to find whether different subsets of T cells have difference in their production, and how much is the production of the T cells in comparison to the production of a nerve terminal?
Yeah, so we did look, so in the mouse, or, so it seems to, your first question, so which subsets have the capacity to upregulate chat and biosynthesize? So So it seems to be pretty context dependent. So in the mouse, a Th2 permissive environment is helping, and some T follicular helper cells are good at producing acetylcholine. And this is in Maureen's science paper in 2019, she details this in quite a nice way. In the human, it appears, we have less data from the human, but it appears that most T cell clusters, as we can call them, I also would agree that that is a better word than subset. So most T cell clusters can upregulate chat. How much acetylcholine they actually make per subset is nothing that we have had the opportunity to measure yet. But they do make substantial amounts. Enough to for example, relax blood vessels ex vivo. For the second question, how much do they make relative to a neuron? I can't really answer that. I guess we should compare that. I'm not clear exactly how to do that experiment. But I think they make sufficient amounts of physiological effects. That's without question. We'll try to find a way, because it's a simple and very good question, and I should know the answer. But I think it's one of these experiments that they are harder to do. This is harder to do than it appears at first glance.
No, it was a naive question because we are used, to quantities, and I was wondering how much an immune cells in comparison to the professional. But, thank you. Number 3.
It's Graham Mackay from the University of Melbourne. It's a related question around, when we think about acetylcholine release, we think of it through synaptic vesicles. Do you have any idea how it's actually being released from the T cells? And also, is there acetylcholine esterase present or regulated in any way?
So the short answer to your first question is no. And we don't know, but we are trying hard to figure this out. But it appears that they may not need the classical synaptic vesicles for release. But it's too early to say, but it's ongoing work. For the second question, is there acetylcholine esterase present? Yes, in massive amounts, which is, it was very fascinating, I think, because the blood or the body is full of acetylcholine esterases, and that's a good thing. Otherwise, we would just be cramping because the muscular end plates would be activated, right? So extrasynaptic acetylcholine is degraded like this.
Yes.
So that's why I was very intrigued when we made these knockout mice that lack the acetylcholine release from the T cells. How could it change blood pressure? Because the blood is full of acetylcholine esterases. And it has to, I don't know the answer, but this is also something I'm very intrigued about. So it's a very good question.
So inside the T cells, do they have acetylcholine esterase to sort of regulate that sort of homeostatic production that might be occurring?
I don't think they have large amounts, although we didn't study that in specific, but outside in the environment, certainly.
Tons. Yeah. Thanks.
Yeah.
So Peter, in terms of the electrical interference, these are very cool data. How deep can you go? In the mouse, you've shown 20 millimeter. In human?
So the physicists tell me that you can basically, depending on the arrangement of electrodes and currents, it's through the what we've done is several centimeters, but they tell me that it's arbitrary.
Okay, perfect. Any more questions from the audience? If not, Peter, thank you very much.
Thank you very much.
And I would like to thank, of course, all of the speakers, Alberto, Peter, Lucy, for the wonderful talk. I think we've been covering a lot of different aspects and how immune response and inflammation is important in different contexts. On behalf of my co-chair, Francesca Levi-Schaffer, and myself, I thank all of you for attending. Just reminding you that this is a session that has been proposed by the Immunopharmacological Committee of IUFAR, and if you are interested, please contact us and you can become a member and contribute to our work. So thank you very much to everyone.
Thank you.
cooking, I burn for you. Hoping one day you'll come home. Can't taste the love on the phone. I love you. I dance in the kitchen on my own. And oh, how we danced through the garden all night. And oh, how we danced away in our bubble. And oh, how we grew each of us hearts and schemes. Home with you now. I dance in the kitchen on my own.
I dip my pinky and lick it, then get to thinking that distance as an ingredient for us could never ever taste right. Mixing some you by my side, I wave the steam from my eyes, and then I realize when I see your smile that you're the prize. When I think of you, you're in the kitchen at the sink with ovens, dancing and singing awake. I dream about our life, family kitchen, forks and knives.
Chop up and reverse, do whatever you want with it, you know?
I dance in the kitchen on my own. Move to the cooking, I burn for you. Hoping one day you'll come home. Can't taste the love on the phone. I love you. I dance in the kitchen on my own. I dance in the kitchen on my own. Move to the cooking, I burn for you. Hoping one day you'll come home. Can't taste the love on the phone. I love you. I dance in the kitchen on my own.
I love you too.
You'll always be my day one, day zero when I was no one. I'm nothing but myself, you are no one else. Thankful you're my day one, thankful you're mine. I got lucky finding you. I won't make the day that I came across you, 'cause when you're with me I don't feel blue. Not a day goes by that I would not redo. Everybody wants to love.
Yeah, that's right.
It's easy when you try hard enough. Yeah, that's right. You'll always be my day one, day zero when I was no one. I'm nothing by myself, you and no one else. Thankful you're my day one. I'm thankful you're my day one. When I first met you, it just felt right. It's like I met a copy of myself that night. I don't believe in fate as such, but we were meant to be together, that's my hunch. Everybody wants true love. It's out there if you look hard enough, enough, enough. You'll always be my day one, day zero when I was no one. I'm nothing by myself, you and no one else. Thankful you're my day one. Hour by hour, minute by minute, I got mad love for you, and you know it. I would never leave you on your own. I just want you to know you'll always be my day one, day zero when I was no one. I'm nothing by myself, you and no one else. Thankful you're my day one. You'll always be my day one. find the love in us. Tell me you can be the one I trust.
So tell me why I should choose to stay when you're old. Keep on treating me in this way. I am stuck and I gave. I don't even want to play. So tell me why I should not just walk away. So tell me why I should choose to stay when you're old. Keep on treating me in this way. I am stuck and I gave. I don't even want to play. So tell me why I should not just walk away. I get so stuck inside my head. This overthinking sends me west. Obsessed about everything you said and what I could have said instead. I get so stuck inside my head. This overthinking sends me west. Obsessed about everything you said and what I could have said instead. I get so stuck inside my head. This overthinking sends me west. Obsessed about everything you said and what I could have said instead. I get so stuck inside my head. This overthinking sends me west. Obsessed about everything you said. So tell me